I'm Lisa from Exosome Diagnostics, and if you are interested in adequately characterizing EV proteins, I would like to introduce you to a semi-automated and super easy way to do that in accordance with MISEV regulations. We developed this new workflow by combining two simple methods. Firstly, the ExoEasy kit from QIAGEN to isolate intact EVs using a bind-wash elute procedure. The EV Elorate offers a range of options for downstream use, and you only need a small amount of it for direct application in the next step, an automated simple Western assay on Jess from Protein Simple. All you need to do is add an antibody of your choice, at least against one target for each of the 3 required MISEV categories, to cover EV positive marker, here in blue, and non-EV marker in orange. The table lists several primary antibodies that have been identified to work on Jess. With this system, you use nice quantitative and size-based data. What that looks like, you can see exemplary with flutalin-1 as target in figure 2 on the left side. A and B show 2 different graphical representations of the signal of this EV protein increasing with the plasma amount used. At the same time, however, the background signal, in this case its IgG, remains the same. These cross-reactions occur from time to time, depending on the antibody, but can easily be identified by a second antibody control, in which the antibody diluent instead of the primary antibody is used. That way, these signals can be identified and excluded from the results. We did input titrations to ensure that our assays have the efficiency and detection range we need. And scaling of protein signal with increasing plasma input can not only be found for flutalin-1, but for all 6 tested EV protein markers, as you can see in figure 3 in blue. Contrary to that, all 3 analyzed non-EV markers that derive from co-isolation during EV elution behave like the IgG background signals and stay at the same level no matter of input volume. In this table, you also find the already experimentally optimized antibody and sample dilution for each assay. By normalization to the highest input volume of 8 ml, we observe similar trend for total protein and EV protein markers, but not for EV negative contaminants containing plasma and lipoproteins. In Figure 5, we analyze different fractions of a plasma isolation to take a closer look at the purity of the ExoEasy isolation. Here we show that EV proteins are not detectable in neat plasma, but can only be visualized after isolation. The EV Elorate shows an enrichment of EV proteins like flutalin-1 while being strongly depleted for non-EV proteins like ApoA1 or albumin. To summarize, we developed an easy and robust workflow for EV protein characterization that fulfills the MISEV criteria for analyzed proteins.